PharmaPal.
You're listening to Bloomscast, the official podcast of Blooms.io, home of the bioactive library for mass spectrometry instant optimization. Let's dive into the latest in natural chemistry, bioactive discovery, and the future of lab innovation.
Today we're jumping into something really cool, how scientists figure out what's useful in nature. We're talking about evaluating natural products.
Yeah, finding those hidden gems from plants, microbes, you name it.
And the methods are just getting incredibly advanced, moving way beyond just, traditional observations.
Absolutely. It's a whole new ballgame.
So our mission today is to get a handle on how someone like a photochemist, really plugged into that whole natural product science world, approaches this, especially finding new medicines.
It's a fascinating perspective. We've always learned from traditional use, but now we can see things at a molecular level like never before.
Okay, so let's start with these omics technologies. That sounds pretty complex. Genomics, proteomics?
It does, but the core idea is getting a complete molecular picture, a snapshot of everything going on inside a cell or organism.
Everything.
Pretty much. So, genomics looks at the genes, maybe finding the ones that help a plant make a specific medicinal compound.
Oh.
And the really powerful part for the natural product community is using computers to wade through all that data.
Right.
We can predict which compounds might actually do something useful, biologically speaking. It's like a super search for drug leads.
So it shortcuts the guesswork. Less trial and error.
Exactly. Big time saver. We look at a compound structure, compare it to what we already know, and make educated guesses about its activity, even potential toxicity.
That must be huge for photochemists, too, dealing with light-sensitive stuff.
Definitely. Knowing what you're looking for or what properties to expect makes handling those delicate compounds much more efficient.
Okay, so say we've found a promising plant or microbe. How do we get the good stuff out, the active bits?
That's where modern extraction comes in. Think high-tech filters. Things like supercritical fluid extraction or microwave-assisted. They're much more precise.
Better than just boiling leaves in water.
Oh, far more effective and selective. We can target and pull out specific bioactive compounds from really complex mixtures.
And once you have that extract, which might still have loads of compounds, how do you find the one molecule doing the heavy lifting?
Good question. That's bioactivity screening. We run specific tests called bioassays.
Okay.
These act like detectors for a certain biological effect. So if we're looking for something that kills bacteria, the bioassay flags the compound or fraction of the extract that actually does it.
Aha moment.
Kinda, yeah. For a photochemist, maybe it's seeing which compound interacts with light in a specific disease-relevant way.
No, I've also heard about network pharmacology. That sounds like zooming out again.
It is. It takes all that omics, data genomics, proteomics, etc., and maps out how a natural product interacts with the whole biological system. Not just one target, but the whole network.
Understanding the ripple effects.
Precisely. It helps the whole natural product community understand the how, not just the what. How does this compound work across different pathways?
And making these natural compounds work better as actual drugs, that involves nanocarriers, little delivery vehicles.
That's right. Putting plant products inside nanoparticles can seriously improve things like stability, how well they dissolve, getting them to the right spot in the body.
Makes sense. Making nature solutions a bit more targeted.
More targeted, more efficient, yeah.
It's really interesting how this blends the old and new. Does traditional knowledge, like from ethnobotany, still play a part?
Oh, absolutely. Ethnobotany is crucial. Documenting how indigenous communities have used plants for centuries gives us vital clues.
Points you in the right direction.
Exactly. It helps the natural product community focus on plants that likely have something bioactive. It's tapping into generations of human observation.
So once you find that promising natural compound, what's next? You have to make more, right?
Yes. Lab synthesis is often needed to confirm the structure and just get enough material to study properly.
Okay.
And importantly, researchers often modify the natural structure, tweak it a bit.
Why do that?
To maybe boost its medicinal effect or reduce unwanted side effects, kind of like optimizing nature's design.
And keeping track of all these compounds, verifying what's what, that requires specific tools. I see things like HPLC, LCMS mentioned a lot.
Yeah, those are key analytical tools. HPLC, LCMS, GCMS, they let us create detailed chemical profiles, like fingerprints, for plants or extracts.
Fingerprints. I like that.
It's vital for quality control, making sure herbal products are what they say they are and contain the right amount of the active stuff. Super important for the whole field.
So putting it all together, these advanced strategies are really pushing the boundaries, letting researchers evaluate natural products much more effectively.
Definitely. We're finding and developing potential new drugs faster and with more understanding than ever before. And for photochemists, understanding that light interaction adds another really exciting layer.
It really does. It leaves you thinking. As we keep merging this deep traditional wisdom with cutting-edge tech, what else is hiding in nature waiting to be discovered?
And how will photochemistry specifically unlock some of those secrets? What are the ethical lines as we get better at modifying these natural blueprints?
Lots to think about.
Thanks for tuning in to Bloom's Cast, brought to you by Blooms.io. Stay curious, stay connected, and we'll see you next time on the frontier of natural product innovation.
PharmaPal.